We are in the seed distribution area for the collection of wheat. In this moment, the staff is preparing the backup for Svalbard, and they preparing 8,000 different accessions. One set is for the backup in Svalbard, and the other 4 sets is for to test germination. One set is for to test in 20 years, and the other set in 30 years, and the other in 40 years. In the final set is for 15 years to preserve in Svalbard. It's only to monitoring the seed viability.
Welcome everybody to the Erie Gene Bank. In our gene bank here, we have categories of storage of germplasm. We have the long-term storage, which is just the room behind you here. This is where we keep our germplasm for a long period of time. And the temperature we use here is -20 degrees centigrade, and they are in freezers. Other gene bank, they are in a room, like what I saw in Seattle and Erie. But here we keep it in freezers because we are thinking more about the health aspect of the workers. You know, if it is kept at that temperature, we think about the health aspect. But we can still think about improving on it. So here we have our base collection and also the MOS collection. They are all in here. In total, we have about 18,600 accessions of more than 300 genera and about 1,700 species. So like I said, this is the base collection. Materials are stored here at a temperature of -20 degrees centigrade. So this is our active collection. We have 2 rooms, and here we try to maintain the temperature of 8 degrees, 5 to 8 degrees temperature. We are still discussing whether that temperature could be retained or we could reduce it, you know, in harmony with other gene banks. Everything is monitored on the computer so that if anything goes wrong, we'll be able to see from the computer. This is the germination room. And this is the germination team. They are doing a lot of germination exercise here, because this is what we do to determine the level of viability. It can give us an indication. The materials we have in the cold storage there, whether they are declining in viability. So we do test at regular intervals, you know.
Greetings, everyone. I'm Yanni Van Been, and on behalf of the Gene Bank platform and the Crop Trust, thank you for joining us for today's Grow webinar. With me is Faye Wambua Ludeling, who is co-moderator for this presentation. Before we get started, a couple of things to note. We have everyone on mute to avoid background noises that may distract you from listening to the webinar. If you have any questions or comments during the presentation, please type them in the Q&A box in the lower part of your Zoom control panel. A recorded version of this webinar, as well as the presentation, will be made available on the GeneBank platform website after this webinar. It's my pleasure to introduce our speaker and longtime friend and advisor to those in seed conservation, Dr. Fiona Hay. Fiona has more than 25 years of experience in seed conservation science. She studied her PhD and then worked in the Seed Conservation Department, later known as the Millennium Seed Bank of the Royal Botanical Gardens, Kew, in the UK. She then spent more than 8 years working for the T.T. Chang Genetic Resource Center at Erie in the Philippines. She is now a senior scientist in the Agroecological Department of Aarhus University in Denmark. Fiona coordinates the seed quality management activity of the CGR Gene Bank platform, and she's also the chief editor of Science, Seed Science and Technology, published by the International Seed Testing Association. She's also a member of ISTA's Moisture Technical Committee. The title of Fiona's presentation today is Seed Survival in Storage, a deep dive into the factors that influence seed longevity. Welcome, Fiona.
Thank you, Niani. Good morning, good afternoon, good evening, wherever you are in the world. And today, as Yanni said, we're going to be talking about seed longevity, and that's seed longevity mainly from the perspective of gene bank management. But before we put our feet into the water, I thought it would be quite useful if we start off with some definitions, and the most important being, of course, seed longevity. So we usually define seed longevity as the length of time that seeds remain alive in a given storage environment. And there are two main storage environments that I'll be mentioning today. First is experimental storage, and this is where the rate of aging of the seeds is accelerated. And then I'll be talking quite a lot about storage in the gene bank. That might be in medium-term storage, and that's where a gene bank usually has the active collection. So this is the collection from which the seeds are distributed. And then there's also long-term storage, and this is where we have the base collection, and this is for long-term conservation. Here is P50, and this is the parameter that we often use as a measure of the longevity of a seed lot. It's the length of time it takes for the viability of a seed lot to fall to 50% in a given storage environment. Whether seeds are alive or not, whether they're viable, is usually determined by a germination test. Sometimes we use another viability test, and the most common is the tetrazolium test. And then for those of you that don't work at a gene bank accession is a sample of plant material that is conserved to represent a particular variety or line or population. And in many seed banks, many seed gene banks, each accession is represented by a sample of seeds in both the active and the base collection, so in both medium-term and long-term storage. So that's some definitions out of the way, and what I really want to cover this afternoon for me, is firstly something about a seed lot as a population of seeds, seed survival curves, and gene bank viability thresholds. Then talk about some of the factors that can influence the subsequent longevity of seeds when they are actually placed into storage. And then lastly, a question about whether or not the current gene bank storage conditions are acceptable, whether or not they're fit for purpose. So first of all, a seed lot as a population of seeds. And when we put a seed lot into storage, each of those seeds hopefully will germinate at the start, but they'll start to age during storage, and they'll start to show signs of aging, and eventually they'll lose the ability to germinate. The actual timing of the loss of the ability to germinate will vary from seed to seed. So what that means is at the start of storage, if we did a germination test, all the seeds should germinate hopefully at the start, and they'd be quite uniform and fast germination. If we took seeds out of storage at different time points and germinated them, germination would gradually get slower and less uniform. It would become more sporadic. We'd start to see abnormal seedlings, and eventually none of the seeds would germinate at all. And that is also illustrated here. So the timing of loss of ability to germinate varies, and this is the distribution. And you can see that the rate at which seeds die is quite slow to start with. It accelerates and we reach a peak, and then it slows down again. And this means that when we do a germination test, we have a sample of seeds in storage, we take them out at different time points and do a germination test. we get a survival curve which usually looks something like this. And this variation in the time when the seeds lose the ability to germinate is actually a good thing for gene bank managers. And that's because it means that we have a bit of a warning. We can see that the seed lot is starting to lose viability. If we had a seed survival curve which looked something like this, we wouldn't know where that catastrophic loss in viability would occur, where it would happen during storage. We really don't want to see seeds going from 100 to 0% viability very quickly. And it's good that there is that variation in the time when the seeds lose the ability to germinate. So here's our typical seed survival curve. And I've added here a line at 85%. And that's because 85% viability is the viability threshold that many gene banks use. So what that means is that most of the seeds in gene bank storage would have viability above 85%. And if a gene bank does a germination test, a viability monitoring test, and finds that the viability is less than 85%, we'll probably go on to regenerate that accession and produce a new sample of seeds to put into storage to replace those old seeds, the old seed lot that has lost viability. I always think 85% viability is a good threshold. And that's because it's after 85%. So when viability has declined to 85%, we start to see this acceleration in the decline in viability on the percentage scale. So this is really quite fast loss of viability now. If we had it any higher, of course, we'd be seeing very much higher rates of rejuvenation. We'd have to be regenerating material more frequently. If we had it any lower, then because they would be on this faster declining part of the survival curve, we'd be at much greater risk of losing genotypes or even accessions. And the other thing to bear in mind here is that the recipients, the people who request material from the gene bank, they might not even be able to get the same germination as the gene bank can. And this might be because of phytosanitary treatments when they request material, or it might just be that they're not germinating the seeds under such good conditions as the gene bank uses. I've got a picture here of the FAO gene bank standards. So these are the standards. These ones were published in 2014. And these standards are what a gene bank should be following. They should be aiming to adopt for their gene bank. And actually the gene bank standards in relation to the viability threshold are a little bit contradictory. In this standard here, it refers to 85% of initial viability, whereas in this one here, it just has the 85%. It does give a let out, so some species, it's acceptable to have a lower viability standard, and that's specified here. And again here, where you might have to have a standard as of 70% or lower. And this is most common for wild species, a lower viability threshold. Just to have a look at that 85% of initial as a viability standard. So I've added it on here if we started off with very high germination and then germination declining. You can see that if we had 85% of initial, we'd actually be ending up regenerating accessions when the viability had fallen to different levels. And we'd probably want to set different viability monitoring intervals. So that's quite complicated from a management point of view. And it also means that if you started here and you were going down here, then you would be already on this steep part of the survival curve. And then, as I said, at greater risk of losing genotypes. There is, however, one case where the 85% of initial might be acceptable. And that's when you have a survival curve which looks something like this. So we did see this for some seed lots at Erie, not very often, but sometimes, where basically the viability is maintained at a high level, not at 100%, but it's maintained at a high level for quite a period before the viability starts to decline. And in a case such as this, the 85% of initial would be okay, because the steep part of the survival curve is down here. Of course, it's difficult to know if you've got a survival curve that's going to be like that. The other, I was going to say, this is an example actually where we saw this pattern, this shape of the survival curve. So this is some real data. It's data for Digitalis purpurea, which is a temperate woodland species. And this was work that was done by one of my PhD students when I was still at Kew, Laura Butler. And this is, we had the seeds, we've stored them at 45 degrees C, so and 60% RH. That was experimental storage throughout. That's this curve here. And again, it's on this panel here. And then the other curves are for seeds that were removed from storage at different time points. And then they were primed, and then they were put back into storage. Now, seed priming is an invigoration treatment where you allow the seeds to rehydrate, but not enough that they actually go on to germinate, but enough that you start the early germination processes so they repair and so on. And what we found here, that when we did that priming treatment, seeds that were still alive, and when we put them back into storage, we did have that plateau phase of the survival curve before the loss in viability here. The other shaped survival curve that we often see is one that looks like this. This is data from Rama Jawad. So she's doing a PhD with Reading University as part of the seed quality management activity of the GeneBank platform. Again, this is experimental storage, and she's working on, in this case, it was seeds of a wild wheat species. And you can see that there is, first of all, an increase in the germination of the seeds at the start of storage before germination then starts to go down. And this is because there is the breaking of dormancy early on in storage. We do see this pattern in GeneBank samples, and I think it just emphasizes the importance of doing a test and assessment of the seeds at the end of a germination test, and especially in the initial test. Just usually it's a cut test, so you just check to see whether the seeds that are still on the plate and haven't germinated, whether they're still fresh and look like they could go on to germinate, or whether they've actually gone soft and rotten away. But making decisions based on just one or a few results from the start of storage can be really quite difficult. So if we had 90%, we wouldn't know perhaps whether we were going to have a survival curve which looked like this, a classical shape, or one where the viability was going to be maintained and then decline, or whether there might actually be some breaking of dormancy. This just emphasizes the importance of experience in terms of managing collections and making decisions. Importance of evaluating the seeds that do not germinate, and often it would be recommended to sample another, take another sample of seeds just to confirm the viability result, and particularly if you get low germination for the initial test. So that was something about the variation in the individual seeds in a population in terms of when they lose the ability to germinate, and seed survival curves, and and gene bank viability thresholds, and why, hopefully you've understood why I think 85% is a good viability threshold. I'm now going on to talk a bit about some of the factors that can impact the subsequent longevity of seeds when they are first placed into gene bank storage. And basically everything that happens before storage can affect the subsequent longevity. And that's in the field, when the seeds are developing, the conditions. It's when they're harvested, the maturity of the seeds and the conditions when they're harvested. It's also what experience during seed processing and duration of processing as well. Just looking at a little bit at seed development now. Don't worry too much about these actual values. What I want you to take away here is just the overall patterns in seed development we see. And here I've got mean seed dry weight, and you can see that the seed dry weight, it's very low to start with, of course, and then it increases, and then at some point it'll plateau off. And the point when the accumulation of dry matter stops, we call that point mass maturity. And then here I've got moisture content, and again, moisture content would be very, very high to start with because the seeds are just bags of water. And then it gradually declines as dry matter is accumulated. And then after mass maturity, the decline in moisture content gets faster as the seeds equilibrate to ambient conditions. And then there are three properties of the seeds that we're interested in. Fresh seed germination. So this is the germination of the seeds if you took them off the plant and immediately put them to germinate. The desiccation tolerance. So if you took the seeds off the plant, you dried them down and then put them to germinate. And then the longevity, which I've got in this line here. So you can see how the ability to germinate and the desiccation tolerance increases during seed development. The relative timings of these varies from species to species, but most species have acquired desiccation tolerance by the time or soon after mass maturity. The really important thing to note here is this development of longevity. And basically longevity, because we're storing seeds in air-dry storage conditions, we only start to accumulate longevity after the seeds are desiccation tolerant. But the most important thing to note is that it continues to improve. The longevity continues to increase really, really late in seed development. And right up until the point of seed dispersal. So what we say for wild species is we always say you should harvest seeds as close as possible to the time of natural seed dispersal, because that's when the longevity of the seeds is likely to be greatest. Many of us are working with shatter-resistant crop species, so the seeds don't disperse. And then in that case, the moisture content of the seeds, as they're sitting on the plant and exposed to ambient conditions, moisture content of the seeds will fluctuate. When it goes down below a certain level, the seeds might be ready to harvest. And we might call that harvest maturity. If it goes up again, as the ambient humidity goes up again, we might delay harvest until they've dried down a bit again. Here you can see the curve for seed longevity. And you can see that here I've got it sort of continuing or declining. And basically this change in longevity late while the seeds are sitting on the plant, pattern can vary from species to species, and it might decline sooner, it might stay up high for longer, depending on the species and depending on the conditions, the ambient conditions that the seeds are exposed to. And that's illustrated here. So this is data we collected when I was at IRI. This is data for 20 Oryza sativa accessions from the Erie Gene Bank. I've got 2 harvested there. The seeds were harvested at 24, 31, 38, and 45 days after flowering, and they were dried in the gene bank drying room. And then we put them into seed storage experiments to get the P50, to get then the measure of longevity. So that's the P50 on this scale, and this is the days after flowering. And you can see, so mass maturity varied between these, the timing of mass maturity varied between these accessions when they were grown here at this time. Some of them had acquired mass maturity already by 24, and some it was a bit later. You can see how longevity is increasing for some accessions, this one quite dramatically. For some accessions, it's going up and then plateauing, and we have some where there's a decline in longevity by the last harvest as well. So the pattern can vary between species, between accessions, genotypes, and it will depend on the environmental conditions. This is also data for rice, and I have to thank Richard Ellis from the University of Reading, who presented this slide at one of our seed quality management workshops. This is data for a japonica rice, which is usually grown in a temperate environment. And they were looking at the effect of growing this accession, this variety, in 2 different conditions. So a cooler environment and a warmer environment, which was meant to simulate the conditions at Baños, at Erie in the Philippines. In the top panel here, you can see the change in seed dry weight. So this is this one. As I showed you in the schematic, it starts low and increases and plateaus. And then here we have the moisture content, which declines during seed development. And this pattern, you can see it didn't differ very much for the seeds produced in the 2 different environments. But when we look at the longevity of the seeds during seed development, so the seeds taken off at different times and then put into a storage experiment, you can see how the longevity increases and then plateaus, but it increases, so it ends up much higher for the seeds that were grown in the cooler environment than for the seeds grown in the warmer environment. So the difference in temperature had no effect on seed filling, but quite a considerable pattern on the considerable effect on the pattern of seed quality development, and by that we mean seed longevity. Another example, again from Richard, to illustrate how the conditions during seed development can influence the longevity of the seeds. This is the data for rapid cycling brassica seeds, and it's looking at the effect of when terminal drought is imposed on the longevity of the seeds. So these seeds down here, these were from plants that were irrigated throughout, whereas here the irrigation was stopped. And you can see how, depending on the timing of when irrigation stopped, it had an influence on how quickly seed longevity improved, and a little bit on the maximum longevity compared with the seeds that were from plants that were irrigated throughout. So that's something about the conditions during seed development and how they can influence the longevity of the seeds when you subsequently put them into storage. The other really important factor is what happens to the seeds after they've been harvested, and in particular, how they're dried. And this is data from Catherine Whitehouse. So she did her PhD with me at Erie and was registered with Richard at the University of Reading. In this slide, we've got the data from 20 Oryza sativa accessions, and they were harvested on different times and between 24 and 48 days after flowering. And after each harvest, the seeds were either dried immediately in the gene bank drying room, So that was at 15% relative humidity and 15 degrees C. Or they were initially dried with heated air in a batch dryer, or before they were transferred to the drying room. After drying, we did seed storage experiments, again to get a measure of longevity as P50. And then we worked out the improvement in longevity, the improvement in P50 for the seeds that were initially dried at higher temperature compared with the seeds that were immediately placed into the gene bank drying room. So this is the percent improvement in longevity, and basically here at 100% means that there's a doubling in the P50, so a doubling in the longevity of the seeds. And we saw a positive effect for 13 of the accessions here. And you can see this one, this was a really, really very significant effect. And the other important thing to note is that treatment didn't have a negative effect on any of the accessions. And you can see that more clearly here. So this is the data from that first experiment, which is this one here. But then she went and did quite a lot of further experiments, and this is a graph from her PhD thesis. And basically what we realized was it wasn't to do with the chronological age of the seeds when they were harvested. It was very much about the moisture content of the seeds at harvest as to whether or not they benefited from initial drying at the higher temperature. And then as you can see, this is just flat here, so we didn't see a negative effect. And as a consequence of Catherine's research, they have now introduced two-phase drying at IRRI, so initial drying at a higher temperature and then final equilibrium drying under the standard conditions of 15% RH and 15 degrees C. We also saw a benefit of drying at a higher temperature for some of the wild species at IRRI. So this was work with Stephen Templey. And you can see here the survival curves for some different species, dried in the gene bank drying room again, or at a higher temperature, initially at a higher temperature. And you can see how initial drying at 30 or 45 degrees C is better for the subsequent longevity of the seeds. We've also seen a potential improvement for some other species, soybean and Bambara groundnut. And this work is continuing again as part of seed quality management activity at IITA, and this is Shola and Tony Topi, who are both doing PhDs. So this sort of raises a question about whether what we do after harvest can compensate for what happens before harvest. And I would say that we really don't know enough about that. But what we do say is that if it's necessary to harvest before harvest maturity, it might be a good idea to hold or dry the seeds under conditions that simulate natural conditions. And we mean natural conditions in a good season. And then you'll have to work out what that good conditions, a good season might actually be. The other thing that I've often wondered about is whether if we had all the conditions perfect for a particular variety, say, whether there was a maximum level of longevity that they could attain. And that would be like their maximum longevity. And that brings us on then to the idea of the genetic basis of seed longevity. And again, this is work at IRRI. This is work that Jae-Sung Lee did as part of his postdoc, a genome-wide association study using 299 indica group Oryza sativa accessions. And the seeds of each of those accessions was harvested at 31, 38, and 45 days after heading. And then they were put into standard seed storage experiments. And those different times of harvests, that's the different colors of the survival curves, which you see, is just some of them in this panel here. You can see, for example, if you look at this one, and these are seeds of the same species, the same variety group. They're stored at exactly the same moisture content and temperature, but you can see that there really are quite big differences in longevity in the survival curves of these different accessions. So this means that we are able to identify genes that might be involved in longevity. And in fact, in that particular study, 8 loci were associated with seed longevity. The other factor that I said was important was the length of time from harvest until the seeds actually get into storage. And this is some viability monitoring data from IRI, from the IRI gene bank. And it's data that I actually downloaded quite a long time ago. It's not been published in this way. These are accessions, seed lots that were regenerated in 1990. And the different graphs here, they're sorted according to the length of time between harvesting and between banking. So seeds here were the ones which went into gene bank storage the fastest, and then you can see that there were quite considerable delays for the seed lots from that particular regeneration year. One of the interesting things you can see in this data is the seeds that were put into storage fastest is perhaps a sign of more dormancy in those seeds. And that's dormancy in those seed lots that was broken perhaps during storage, or you can perhaps get an idea that it would have been broken if they were just sitting in the drying room, which is where the seeds were usually kept before they were processed. You can see the distribution of the germination gets higher for these groups here for the initial germination test. But really, most worrying is what you see here is that the seed lots which were later to get into gene bank storage, so a longer period between harvest and storage, they are perhaps showing more decline in viability, a faster rate of loss of viability compared with those that made it into gene bank storage the fastest. So variation in seed lot longevity within a species. So that might be due to genetics, the conditions during seed development, the conditions at and the timing of seed harvest, how the seeds are processed, the drying and the holding conditions, and the overall length of time before seeds are placed into storage. And we know really very little about how all these factors interact to affect seed longevity. And because of that, we basically still need to monitor the viability of seed lots in gene bank storage. So that brings me on to the final part of my presentation. And I've posed that as a question. Are current gene bank storage conditions acceptable or not? Are they fit for purpose? And I'm going to try and have a look at that from some of the work that we've been doing, looking at the historical viability monitoring data from some of the CGIAR gene banks. Again, these are the FAO standards, and this is what they say about storage conditions. The seed samples should be dried, and then they should be sealed inside a suitable airtight container. And then for the collection, long-term storage, they should be at -18. And then for the medium-term storage, the active collection can be stored at a higher temperature at 5 to 10 degrees C. And even though I've said that we can see quite a lot of variation in the longevity of different seed lots within a species, we can nonetheless say that a seed lot of a particular species is likely to be relatively long-lived or relatively short-lived. And this is what this graph here is illustrating. These are predictions of longevity, predicted times of viability fall from 98 to 85%, so to a viability threshold. If the seeds are stored under long-term gene bank storage conditions, so in this case, -20 after drying to 50 equilibrium with 15% RH and 15 degrees C. And you can see, if you look on this scale, there is a huge variation across species in their relative longevity. And we get some species which are really very, very short-lived. And this is another species that we worked on when I was at Kew. And these seeds of this species would die in gene bank storage within a year or two. A couple of other species just to point out here, we have groundnut, Arachis hypergaea. We have soybean here, we have Oryza sativa rice here. And then over on this end, we have some seeds of some species which are predicted to be really extremely long-lived. And that is something that's also reflected in this graph here. So this graph was kindly shared with me by Louise Colville. It's from their publication in New Phytologist. And here they did a meta-analysis of the longevity of different seeds of different species in different storage environments. So they took data from some different publications. Experimental storage conditions, ambient temperate conditions, or in gene bank data. And this was data from the USDA Gene Bank, the base collection at Fort Collins, which was published by Christina Walters and co-authors. And what the point that they were trying to make in this paper in particular was that when we look across diverse species, we find that most species have significantly poorer seed longevity than the maximum observed for the different storage environments. So most species are grouped down at this end rather than up towards this end. But these, just focusing on these ones in particular, I mean, we have longevities here. So this is estimates based on extrapolation of data, and you can see there must be extrapolation of data because we have not been storing seeds for 400, 500, 600, 600 years. And that's just one of the problems in trying to understand whether the gene bank conditions are good or not, is that we haven't been storing seeds for very long relative to longevities that we think we might be able to get. So looking at the CGIAR historical viability monitoring data, so we started this work a few years ago. So this is data that we downloaded from the Gene Bank information management systems going back a few years. This is the data for seeds in medium-term storage, and each graph is for a different collection. So a different species and gene bank. Some of these are grouped species. Along the x-axis, we have the estimated storage period, which is going up to 32.5 to 37.5 years. So that's the maximum estimated storage period that we had data for. The green bars, they're the proportion of results within all the results for a particular collection. It's the proportion of results that were above 85%. And then the red bars are the proportion of results where the viability was less than 85%. You can see perhaps in particular down here, this is data for the tree species seeds at the World Agroforestry Centre, where we have got more of the red bars. And this is just perhaps illustration of the lack of investment into the ex situ conservation of tree species. Same type of graphs for seeds in long-term storage. And now you can really start to see that we are actually getting quite a lot of seed lots where we do see viability maintained for 30 years or more. So looking at some of that data in a more classical way, so this is data for Oryza sativa from both Africa rice and Erie, and again, medium-term storage and long-term storage. And so you can see certainly in the lower two graphs, you can see how quite a significant proportion of seedlots are making it, still have high viability after the 30 years or more in storage. And results that we've seen from the CGIAR gene banks are favorable compared with other studies. And in particular here, I've looked at the results from Christina Walters' study, which was the study that I mentioned when I showed you the graph from Louise. So what I've added on, so this is the same graphs that you saw in the previous slide, but what I've added on is the results from the Walters paper, where they have a mean initial germination, the mean final germination, and you can see the storage time there, and also a P50. And because the seeds at the USDA-based collection, they were stored both initially at 5 degrees C, and then they were moved to minus 18 degrees C. I've put those values on both the MTS and the long-term storage for our data. But hopefully you can see that we do seem to be doing okay. It's always a bit hard to know because so my screen keeps freezing, so I can't move the pointer at the moment. But if you look at the Erie graphs, so any seedlot that gets below the viability threshold, so when viability falls below 85%, those seedlots wouldn't normally continue to be monitored. So that means that the results that we have for the latest storage periods, they are the seeds that are living longer anyway, if you like. So it's very hard, again, to get true estimates of longevity or of P50 or whatever. The CGIAR viability monitoring data has also highlighted where we do have some problems, and here this is the data for groundnut from ICRISAT, and also for some of the Arachis forage species stored in the CIAT gene bank. And you can see in the MTS for the seeds in ICRISAT, you know, that's quite a long storage period, but we are seeing quite variable germination. It's better for the long-term storage, but we are seeing some sign of decline in viability after 20 years in long-term storage for the groundnut. And for the wild species, the forage species here from CIAT, you can see there are some data there where after quite a short storage periods, we might be wondering what the viability is. So to answer the question, are current gene bank storage conditions okay based on the CGIAR viability monitoring data? I think we can say that they're certainly maintaining the viability well for most crop species. There are quite high rates of regeneration at some of the CGIAR gene banks. And given the predicted and the achieved longevity of seeds of these species, this must be mainly due to low seed number. And there are various reasons why the seed numbers might be low. So there does need to be more effort to optimize the quantity of seeds produced and placed into storage. And so some of the reasons might be that seedlots don't meet phytosanitary conditions. So that means that a seedlot has to be replaced, or you only get a few seeds that you can actually put into storage. For some species, and Oryza might be an example, because we expect those to be relatively short-lived, because they are relatively short-lived compared with some of the other species, we may be starting to approach steady state gene bank operations, but In general, I think most gene banks are really still too young for us to be getting to that point. It is really, really important that where we can, we continue to monitor the viability of older seed lots, and perhaps with even shorter monitoring intervals to get a better understanding of longevity in gene bank storage, and so that we're able to be more efficient about viability monitoring in general, and in understanding expectations about regeneration needs. So I think we're doing well, but we have a lot more to learn about seed longevity in gene bank storage and the factors that influence longevity if we're going to further optimize gene bank operations. And with that, I'd like to say thank you to the staff of the CGIAR gene banks with whom I collaborate through the seed quality management activity of the CGIAR gene bank platform. And in particular, I would like to mention Marie-Noël from Africa Rice, Charlotte at the Crop Trust, Jason and Stephen, who I worked with at IRRI, Catherine, who I also worked with at IRRI, who's now at the Australian Grains Gene Bank, and Richard Ellis from the University of Reading. And thank you to you for joining the webinar.
Thank you very much, Fiona. That was a wonderful walk through seed lot viability to the new protocol, the two-phase drying protocol, to your critical look at current storage conditions in the CGIAR. So currently we have 11 questions, and I'll just read them out to you so you have some time to think and go over. Our first one is from Norma Manrique. She's referring to that slide right before Rama, the slide that Rama showed up about, there's a publication there you were referring to, and she asks, this is in an accelerated aging experiment, is it? That was her question.
Yes, so I think it's the graph where we had germination increasing and then declining. And yes, those seeds were placed into experimental storage conditions. And we've been trying to use the same conditions, in different studies so that we can make more comparisons across species and varieties and so on. So we're usually aging seeds at 60% relative humidity and 45 degrees C. And that means that we can get data about the longevity of the seeds within, usually depending on the species, but usually within a couple of months, let's say. And we usually use those experiments to see whether a treatment that we've applied after harvest, say, whether that has an effect on the longevity of the seeds. And that's something that Rama is looking at ICARDA.
Okay. Next question is from Theo Van Hintem. Do you agree that viability thresholds should be defined in combination with the minimum number of seeds tested? A level of 85% with 200 seeds would mean that you want to be 95% sure that the viability is above 80.8. To reach that level with 50 seeds, you would need a threshold of 90%. So he's talking about the tolerances given the number of seeds that you have.
Yeah, yes, I think that is a good idea. I think you can either do that sort of formally, or you can just accept that you have got sampling error there. You're only ever going to be getting an estimate, and if you're always testing the same number of seeds, does that matter in a way, because you've got the same sort of tolerance there, regardless. Of course, you could always say the more seeds you test, the better. But the gene banks, they don't want to waste seeds on germination tests. And some of the other things that we've looked at a bit, and in particular, Luis Santos at CRT, he's looking at sequential sampling, which is when you take out one sample of seeds and test it, and depending on the result, you do another test on another sample of seeds or not. And it's about balancing how many seeds you use, the efficiency and the accuracy of the result.
Yeah, thank you. We have a question from an anonymous attendee. How useful is the Q seed information database in Q? Yeah. So in your opinion, is
In my opinion, it's very useful. So the Q seed information database has different modules within it. One thing it has is just a whole load of information about germination requirements, and these are the results from Q's testing, germination tests. So that's a great resource, especially if you're working on a novel species that you haven't worked on before, just to go in and have a look there. I think one of the other modules in the information database, which might be more what this question is asking about, is the viability module. And this is where we have mainly the viability equations, so the Ellis and Roberts viability equations formulated in a way where you can get out predictions of longevity for certain species depending on the moisture content and the temperature that you're storing them. And I always think that's very useful for illustration purposes. So it can give you an idea of what the viability of your seeds might be if you left them, you know, in your kitchen, or if you left them outside somewhere. So yeah, I'm a fan.
Wonderful. We have a question from Manuela Nagel or Nagel. I'm sorry if I mispronounced that. Dear Fiona, thank you very much for your clear presentation. While analyzing germination results of accessions from long-term storage of IPK gene bank, I often found that the effects of dormancy, release, and increased aging influence each other. Effects of dormancy, release, and increased aging influence each other. I think this interferes with the correct calculation of P50. Do you think that it would be useful to exclude this effect? If yes, do you have any idea how to exclude the effect of dormancy when calculating longevity?
Yes, I'll say yes. Yes, it would confound it, and I think it goes back then to what I said previously, is that it is important to do an evaluation of the seeds that don't germinate at the end of a germination test, so that we can get an idea of whether the seeds aren't germinating, whether that's due to dormancy or not. So I think, yes, if you did that, then you might be able to take that into account when you are doing the modeling to try and get your estimates of longevity.
Here's a short answer. What is the longest lived seed that you have come across?
There are some examples in the literature of extremely long-lived seeds. One is date. I think that's the oldest one, and I can't remember right now, but you're talking thousands of years, I think, so many hundreds of years. Another one was sacred lotus seeds, but if you do a search in the literature, you'll come across the papers for those. I think the oldest one was the sacred, was the date seeds.
A question from Sakayo. Hi Fiona, you talked about the cut test to check non-germinated seed species in wild species. How about the tetrazolium test, TZ test?
Yes, so a tetrazolium test can be used. I think there are some drawbacks with the tetrazolium test, and I mean that might be to do with not having an optimized tetrazolium test. But if you're seeing, if you do a tetrazolium test and you see some differentiation, so signs that some seeds aren't viable according to the tetrazolium stain, and other seeds that are, then maybe you can be confident with that. And then if you use that to compare against your germination results, and especially if you do a cut test at the end of your germination test, then I think the tetrazolium test can be very complementary in terms of truly understanding the viability of a seed lot. And it is useful for those species where we're not quite sure how to germinate the seeds. So yes, it's useful. One of the drawbacks, of course, is that if you're only using a tetrazolium test, and if that's because you don't know how to germinate the seeds, then how is anybody else going to be able to germinate the seeds? So I think we need to make sure that we can, as far as possible, germinate seeds, and don't just use tetrasolium.
Okay, thank you. A question from Okun Unu. What are the molecular mechanisms and epigenetic dynamics of seed development and their implications on seed dormancy and longevity and germination?
And that is a huge, huge topic in itself, and I won't begin to answer it here. Again, I would say look in the literature. You'll find lots of interesting work. Perhaps have a look at papers by Julia Buetink and Olivier Leprance. Have a look at some of their work and go from there, basically. But it is a huge topic in itself.
How many gene banks are using sequential viability testing procedures to save difficult to produce seeds?
As far as we know, as far as I mean, certainly none of the CGIR gene banks have been routinely using sequential sampling. They have been working on it a lot at CIAT. So I think if any gene bank does start to use it, it will be the CIAT gene bank. And I think they see it as quite a promising way to reduce the number of seeds tested and save resources that way.
Okay. So we have another one. How applicable is the data to wild seed lots that you have presented today?
I don't know which particular data, but in general, most of what I covered is applicable to wild seed lots, but I just say it tends to be a whole lot harder for wild seed lots.
Okay. Have you used any non-destructive spectroscopic techniques for testing seed viability?
I wouldn't say for testing seed viability. We have looked at multispectral image analysis for predicting viability. So then that's just based on correlations. And we've only done really just a little bit of work in that area. I think other seed scientists are trying different spectroscopic techniques there.
Wonderful. Is the oxygen level taken into account for the gene bank storage? Meaning inside, I suppose, the aluminum foil package. Or maybe it's in long-term storage air. Is oxygen taken into consideration?
So what we know from seed storage experiments that are done in a closed environment, so usually in aluminium foil packets, compared with seeds that are in an open environment where they have plenty of excess air and oxygen, is that there is a damaging effect of the oxygen, which increases the lower the moisture content of the seeds. So when we're packing seeds for storage, we always advise that you try and sort of squeeze as much of the air out as possible. So just basically make sure that the seeds fill as much of the space as possible. So you minimize the air before you seal them. Some gene banks do use vacuum packing and completely remove all the air. Another option might be to flush with nitrogen, say. The problem then is if you're a very active gene bank and actively distributing materials, then it just adds another step when we're resealing and might take more time or more equipment and resources. And we always say the most important thing is to reduce the moisture content of the seeds, and that's far more important than the oxygen, the benefit of drying.
Next question is, dear Fiona, thank you for your presentation. Very clear, informative, and useful for our work. My question is about the importance of seed drying for seed longevity and which would be the best condition and timing to do it. So a little bit back to what you presented, and I think they want to just pinpoint what you think is the best timing for drying and the best conditions.
I think it goes back to the graph I showed, and I made the point that ideally just dry, or if the seeds are very immature, hold the seeds. Keep them in a humid environment for a while, but then basically dry them in conditions as much as possible that simulate what they might experience in nature, if you like. And I think it's quite interesting, the work on rice, because obviously rice is grown in tropical environments, and they never experience 15 degrees C normally, not the temperate rices. So putting the seeds from the field in at 15, that's, you can sort of imagine that might not be ideal. So I think the advice would be to try and simulate, mimic natural conditions as much as possible, certainly in terms of temperature.
Wonderful. Dear Fiona, do you know of studies on how to interpret aging signs as we see in seedlings, which do germinate and develop into plants, but show signs of chlorosis?
I think perhaps one of the best resources there is actually the ISTA rules, when they talk about the germination test and what counts as a normal versus an abnormal seedling. And for different species, so there you can see the typical abnormalities that you see for different species. We've got some experiments running on carrot seeds at the moment, and we can quite obviously now see what is an abnormal seedling, and the root splits quite early on, and yeah, we see chlorosis as well. So I think that would be the resource I would suggest, the ISTA germination chapter in the rules.
Okay. Is in vitro embryo rescue, this method, is it possible to increase the viability?
You can't increase the viability, but you can rescue embryos which are in a seed which might not otherwise germinate. So I mean, sort of what it says in the name, really. It is a rescue method, when you can't germinate the seeds.
Okay. Here's one on viability monitoring. It says viability monitoring requires much seed and funds. Your prediction on longevity slide showed expected survival of thousands of years. Shouldn't we stop viability monitoring for the long-lived species to avoid unnecessary regeneration and waste of funds?
Yeah. So for some of the species that we've looked at from the CGIAR data, I think we can conclude that for fresh seed lots, as long as the initial viability is high, and I think it's always important still to do that initial viability test. But if the initial viability is high, then you don't have to start testing every 5 or every 10 years. You can wait longer. But I would still always go on the evidence that we've got so far. So if the maximum length of time we've been storing seeds is 30 years, then I would say, maybe you can safely wait 25 years or something. But I wouldn't want to be going beyond what we've actually stored seeds, the length of time that we've actually already stored seeds for. And then, the other point that I made was for those oldest seed lots which are in storage now, I think it's still important that we do monitor those, that we can get a better idea of the actual longevities that we can achieve and see how well they match up with the predictions.
Mm-hmm. Here about statistics, how can standard error be used to explain variations of P50 values among accessions studied for long-term storage experiments or seed longevity?
So you can get, well, so when we're fitting a survival curve, what we usually use is something that's called probit analysis. And that's basically fitting the negative cumulative normal distribution to the germination data. And that probit analysis, it takes into account the binomial error that we expect for each germination test. So the binomial error that depends on how many seeds we're actually testing. And then we can get a standard error when we do that fitting, that modeling process, and get out the P50. We can also get a standard error for that P50 value. So that is one way in which we can then compare those P50 values to see if they're different and if we're then seeing differences in longevity. But the other way we do it is actually to see if we can fit one line through the data if we're comparing different seed lots, say. And that's another way to understand whether there are significant differences in the longevity of different seed lots. So, yeah.
Okay. We have a question from William Solano. Thanks for the presentation. Do you have knowledge about the possibility of predicting viability and vigor of the seed through the analysis of images, area, color, imbibition, things like that?
I think ordinary image analysis on the whole, no, except for imbibition. So there was a paper recently actually in Seed Science and Technology where they had looked at the imbibition process of seeds using image analysis, because when the seeds take up water, they do swell. But in terms of viability, then ordinary image analysis, we don't expect to be very helpful to us. And that's why this relates back to the previous question about spectroscopic techniques to predict viability, which probably has more promise, but it will only ever be correlations because these techniques are looking at the surface of the seeds and not at the embryo, which is the bit that needs to actually grow.
Okay. Komlan Arnoud Gouda, thanks for the presentation. What is the best way and material to use to measure the moisture content in a seed before conservation?
Well, but because we don't want to use seeds unnecessarily, many gene banks now are using water activity meters, which basically measure the humidity of the air around the seeds, and we call that the equilibrium relative humidity. And if you've been drying seeds in a particular environment, then if you measure the humidity, if the RH, the ERH of those seeds is the same RH as the environment, then you know that the seeds have dried to equilibrium. So that's very useful and non-destructive. So that's kind of our preferred method.
Okay. Thank you for your very nice presentation. Do you have a working definition of short-lived seed in terms of how quickly they decline in conventional seed bank storage and could perhaps benefit from cryopreservation? So definition of short-lived seed that would be used in cryo.
Yeah, I don't. I know they were looking at this at Kew, the Millennium Seed Bank Partnership. So they were wanting to identify some of the species which have really, really short-lived seeds. And I mentioned anemone earlier, as the seeds not staying alive for a couple of years at best. So really putting them into conventional storage isn't going to be much good. And they would be a prime example of something which might benefit from storing under cryo conditions. And what Hugh was looking at, and I can't remember the details, but basically when you put the seeds into a standard storage experiment, depending on the P50 from that standard conditions, that would be used to decide whether that species is one that should be prioritized for cryopreservation. Yeah.
Okay. And I think we have the last question. Thanks, Fiona, for staying on with us. What is the information that you need to create ahead of time before you store a new wild species under standard gene bank conditions? I'm interpreting the text. I think that's what they're asking. What information would you have to have about a new wild species prior to putting it under standard gene bank conditions?
I think the obvious one there is an understanding of the likely storage behavior. So obviously everything I've been talking about relates to orthodox seeds which tolerate desiccation, but there are recalcitrant species out there, intermediate species with seeds that don't tolerate that drying step. So I think that would be the most important thing that you need to know. And otherwise, I think, I mean, other things are nice to know, but you don't need to know necessarily. You could put them into storage as a way of conserving, just like that. Yeah. Obviously, it's useful to know the germination behavior. You might want an idea of longevity, but yeah. So the most important thing would be seed storage behavior, the category.
Right. Okay. A couple of people have asked for some of the publications that you have mentioned, and we would just like to say that Fiona has provided her email within her talk. We're going to share that with you after the webinar. So please feel free to contact her if you would like information on some of the publications that she's mentioned. We would like to thank Fiona very much. Also my co-host, Faye, for jumping in and correcting everything, and Melissa for standing by, and all the people at the Crop Trust. Thank you, Fiona, for another very brilliant, very nice presentation. To all of you, we will see you next month. Thank you for attending and for all your questions. People are sending you many regards, Fiona. Thank you very much for that on the chats. I hope you can see that. We'll see you next month. Until then, stay safe and keep well. Bye-bye. Bye, everybody.
Bye. Thank you.